Rosita.
I think he doesn't need to.
Too much presentation at least.
Don't repeat that?
For me, Claudio, he's going to talk about, well, neurons.
Thank you, Rosa, for your kind introduction. It's a pleasure for me to be here. Well, as you know, from time to time, the people that work here want to talk about what we are doing because there are new students and new people coming around. So I think it's important that the people know what the different groups that do here. And what I'm going to tell you today is something that I call reconciling information directionality with a negative time lag in neural circuit, but it's mainly what Fernanda Matias has been doing for her PhD. That was a collaboration with Mauro Copelli from Recife. Fernanda is already in Maceió in Brazil with a permanent position there. Pedro Carelli works in Recife together with Mauro. And there were two other collaborators, Leonardo Lira Goyo, who was a former student of mine here at IFISC, which is now in the Queensland Institute of Medical Research in Australia, and also Steve Bressler, who is from the Center for Complex Systems in Florida, in Atlantic University. So the point is that when we try to record information from the brain, we want to know how the brain works, we have to do some measurement on the activity on the brain. And probably one of the most interesting measurement is the electrophysiological recordings. For that, you have to implant electrodes to get the signal that is generated by the neurons or the cortical areas or whatever. This technique is very useful because it has a very good signal-to-noise ratio, has a high temporal resolution, and you can place the electrodes in precise location. However, this is an invasive technique and can be applied only in animals, or mainly in animals like rats, cats, monkeys, and some others. They are being used also in humans, specifically in people with epilepsy. When they try to find the focus of epilepsy, it is very useful to use this kind of electrode because they permit them to know exactly or with more precision before the surgery where the part that has to be removed from the focus from the epilepsy. Up to now, the drawback of this technique is that you cannot place too many electrodes. Actually, more or less 60 electrodes can be placed together, but they promise to have arrays of around hundreds of electrodes in the near future, so it will be a very dense recording from the brain. Once you have this kind of data, you can do some analysis, or you have to do some analysis, and typically correlation, mutual information, transfer entropy, and some other indicators are being used to analyze the data. The techniques permit the computation of the functional connectivity, but also the effective connectivity, which gives you information about how the information flows from one side to the other. But in some cases, you can also get anatomical connectivity from the brain. It has some advantage with respect to others, non-invasive techniques that include EEG, which is placed here, or MEG, which is placed here, because these are taken from outside the head, and the signal is much more contaminated, it's much more noisy. It has also some advantage, but certainly in humans, these are those techniques that are mainly used. But at some point, we got some data from the group of Steve Bressler in the United States, where they were measuring the synchronization of networks oscillating in the beta band, which is between 10 to 20 hertz. And they try to find the influence revealed by the causality measurement in this area. So they had the monkeys doing what is called a go-no-go visual discrimination task. The monkey is pressing a lever and is seeing some pattern, and at the time the pattern appears, it has to release this lever. If he does that in less than 500 milliseconds after the stimulus appears, he got a reward. Some years later, in 2012, they did a similar experiment, but they studied the frontoparietal synchronization, and this was during a visual working memory task. That means that you have, for 100 milliseconds, you have a pre-sample screen, then you put a sample for 500 milliseconds, then you remove it for a delay time of 800 to 1200 milliseconds, and then you include a perturbing image here, and the monkey has to match the previous image. Interestingly, this is what the electrodes were placed.
What do you mean by to match? How do they match?
They have to choose the right one. With the moving a lever, they have to choose the right cues. This is more or less the schedule of the brain of the monkey and where the electrodes were placed, and this represents here the connections between the different areas. I'm not putting too much information now because I'm going to come back later. But the width of the arrows will indicate the strength of the connection between the sites. And then there are some arrows also that will indicate the influence, which area influences which other. So for instance here, there is a single or one directional influence from area 2 to area 1. And this area belongs to the somatosensory part and the motor cortex part of the monkeys. Interestingly, they conclude that there were strong causal influences observed from the primary somatosensory cortex to both motor cortex and inferior parietal cortex. However, the causal relations were generally inconsistent with the time delay values, which means that when they measure the activity from this side to this one, the information flow was against the time it takes the information to go from one point to the other. So the sign of the time delay did not predict the causal direction. And they conclude, although they didn't enter in more details because in my opinion they didn't understand very well what was going on there, and it's still not very clear. But what they conclude is that the relative phase, which allows you to calculate the time delay, is not a reliable index of neuronal influence. And we agree with that. If you only have as information the time delay or the relative phase of the activation of two sites, you cannot conclude how the information flows. That is true. However, those remember us, some work with this with Raúl Toral some years ago about anticipated synchronization. Because what you usually expect when you measure two sites is a situation in which you have some site that activates, it transfers information to the other side, and the other side activates after. However, what they are telling us is a different story. Their story is the following. You have two sites that connect in one direction, but the receiver side activates before the sender does. And this is what we know from the past that was called anticipated synchronization. So this system is synchronized to the future state of this one. So the question is now, can we predict or anticipate the future of a system? Well, as Raúl used to say, if you give me the equation of motion and the initial condition for the system, of course I can anticipate how the system is going to behave. And for instance, I can tell you that the next total eclipse will be March 20, 2015. It will be visible from Iceland, Europe, North Africa, and Northern Asia. And you know that I'm right. That will be the situation. But we also know that, especially here, we also know that many times we have to deal with fast, varying, or even chaotic systems for which the initial conditions are not well known. And in that case, I'm not able to predict for a long time. However, in the year 2000, Henning Voss, a German scientist, proposed a novel method to predict the response of a dynamical system based on the use of an auxiliary system. The prediction is done by anticipating the evolution of the system of interest. So this looks very similar to what has been observed in the experiment. But let's go to simply the simplest case we know from the beginning when we study synchronized systems. We have two systems, x and y, that are coupled through a given function. The evolution of x is given by the function f. The evolution of y is given by the same function y. And I couple them in a diffusive way, kx minus y of t. So this is immediately obvious that the x of t equal to y of t is a solution of the system. The difference is a fixed point. And this solution is stable for large enough kappa. That we know is very simple to analyze. But what happens when we have delay? I'm sorry, and this is true even for chaotic system, because I'm not saying anything about how is this function f. I'm just coupling this system to a clone system. But the idea of both was slightly different. The anticipated synchronization, the slave system predicts the dynamics of the master system. And he proposed two coupling schemes for which this anticipated synchronization occurs. The first one is called complete replacement. And the variable x is ruled by a decay term here, plus a self-feedback given by a function f of x of t minus tau. And then I couple to y this function f, but at the time 0, at tau equal to 0, instantaneously. So the evolution for y is minus a times y plus the input from x. The second scheme he proposed was slightly different. I have a given autonomous system x, whose equation is ruled by this differential equation here, and it couples to y, but then y has also a delayed term, which is of the form k y of t minus 2. So, yes?
Is this negative kappa? Or?
Yes. Well, kappa is positive, but you have a negative, you have a, let's say, a negative feedback. So now, if we analyze this equation, we find some interesting aspect. F of x is a function which defines the autonomous dynamic of the system under consideration. That describes its own method. In both schemes, the manifold y of t equal to x of t plus tau, which means the slave is doing what the master is going to do in the future, is a solution of the equation, and both show that this can be structurally stable. This is more remarkable when we talk about the dynamics of intrinsically unpredictable system that can be chaotic system, and I will show you some examples in the next transparency. In the complete replacement scheme, for which I'm not going to talk about in the next of the talk, tau can be arbitrarily large, while in the delay coupling scheme, in which I'm interested on, there are some constraints on the values of the coupling and on the values of the delay. And these are some examples of Bose that he published in the year 2000. This is the Ikeda equation. You see x dot equal to minus ax. And you have here as feedback term with a sine function here, and this sine function of x also enters in the receiver side, and this is in solid line you have the evolution of the master, in dotted line you have the evolution of the slave, and you clearly see that the slave is anticipating the dynamic of the master. If you plot x of t as a function of y of t minus tau, you see a straight line indicating that the receiver is predicting what the master is going to do in the future. And then he proved that in electronic circuit. Here you see, of course it's not perfect because you have certain parameter mismatches, but you see that when you take x of t or you plot x of t and y of t minus tau in the same axis versus time, you see a remarkable coincidence between the two trajectories. Also at that time, Cristina Massoyer studied the idea of a laser because we knew that a laser with feedback becomes unstable when you have a mirror in front, and then the output power, the intensity of the laser light becomes erratic. And then if you couple to a receiver system, which is this one, then you will see that for appropriate parameters, you can anticipate perfectly the dynamics of this system. Later on, this was proved experimentally by Jun Liu in the United States, where if you shift already the time series in the slave with respect to the master, you see a very nice coincidence. And also that the time lag you anticipate is equal to the delay time in the master system. Which means that this has been proven, but this is also the complete replacement scheme, in which I'm not so interested. At that time, Raul and I started to be interested in applying these ideas to neuronal systems. But we knew almost nothing about neurons, so we said, what can we use? So we said, okay, let us take a model of equation that describes neuron dynamics. And we choose the IFISC-Unagumo model that is known to be quite adequate for some neurons in some situations. And let us couple as Bose suggested, which means I put a diffusive coupling, which means instantaneous coupling with the master system and a delayed self-feedback, negative one. But now you see that the system is not autonomous anymore. I have an external forcing here. And with this external forcing, the solution x of t, y of t equal to x of t plus tau is not a solution of the system anymore. So this was the idea we had. We had an external stimulus that can be anything, noise, chaos, constant, whatever you want. You feed the master system, you feed the slave system, and the slave has also a delay, negative feedback. Even though the solution of the system doesn't exist anymore, the system tends to operate in the anticipated synchronization solution for some range of parameters. For instance, if this is the coupling and this is the delay, all this region we show here is a delay, is a region of anticipated synchronization, which means that this system can anticipate the dynamics of this system. Although in this situation, this Y is a noisy input, being the same for both. Well, you say, well, this is very interesting because if we can predict what the system is going to do in the future, we can use this as a control technique. So let's build the following circuit. I have the master, I have the slave, I have the negative feedback, I couple them, and if I integrate the equations, I get, for a given, this was the Adler system, I get pulses, and I get in the slave system the anticipate pulses. So I said, well, if I detect in the slave an anticipate pulse, I can operate on the master and cancel the next one. This is a representation of this in the oscilloscope, where we built an electronic version of the system, and now we use a control signal. Now when we detect a pulse here, we will try to cancel the next one in the master. And that's what we did. As soon as we detect this, we send a control signal, and we put a perturbation here that controls the output of the master. And then you see here in the experiment that the slave does the pulse, but the master doesn't do any pulse. So with this anticipated synchronization scheme, we are able to control a system. Certainly, to know very well the equations of the master because I have to copy almost it. Well, this was very interesting, but this has nothing to do with real neurons. So can we apply this idea to the neurons? So some years ago with Mauro and Fernanda, we decided to revisit anticipated synchronization in coupled systems, especially in coupled neurons. So how do we model neurons? Certainly we have to be more careful with the neurons. What we know for neurons is that neurons have a membrane that separates the internal part of the neuron with the external part of the neuron. That outside I have certain ions, inside I have some others, and in the rest the membrane is a potential of minus 70 millivolts. So on the rest, the neuron has a difference in the potential between the external and internal part of minus 70 millivolts. But as soon as I apply an external current, I can start to produce movement of charges from outside to inside and from inside to outside. And this is here what you see. I applied an injection current, and there are some gates that open that allow sodium ions that are positive to come in. Since they come in, now the potential is going to grow. It becomes to be positive, positive, positive. At some point it reaches a maximum value. Then this channel is closed, and some other channels that allow potassium to leave from inside to outside are open. So I have an exchange of ions from outside to inside that generate an action potential in the neuron. And this action potential travels along the neuron and is going to affect other neurons in the brain or in the network. So how do we model that effect? Well, it's quite easy. It is traditionally to represent the properties of the membrane in terms of equivalent circuit, because this is electric. According to the Kirchhoff law, the total current that flows into this circuit that contains sodium ions, potassium ion, and other ions that I'm just counting as a leakage current. And I also have a capacitor here. So if I put the equation, it's very simple. The total current is the current through the capacitor plus the sodium current plus the leakage. Or if I rewrite it, I have a differential equation for the evolution of the voltage across the membrane, which contains the current, the sodium current, the potassium current, and the leakage current. And if you see here, the conductance, or the inverse of the resistance, does depend on the voltage, because we know that for certain voltage the channels are open and for others are closed. So this conductance will change with the voltage across the membrane. And how does that happen? Because these channels that act as doors or gates between the internal and external part of the membrane can be closed and not activated, can be open and activated, or can be open but inactivated. So there are two mechanisms that prevent charges to pass by the channel. It can be either closed or can be either blocked. The probability the channel to transmit charges through it is given by the probability that the channel is open and depends on the number of channels I have, and the probability that is activated and the number of channels that are activated. So assuming that all these units are independent, I can write the probability as this term. And then how I model these probabilities, how the probabilities change in time. Well, I use a first order differential equation, which gives me the rate of opening of channels times the probability to be closed, minus the rate to close times the probability to be open. So this is a very simple equation for the rate. So with this in mind, Hodgkin and Huxley in the year 1950 developed a model that considered three currents: potassium and leakage current. This is related to the potassium channels, this is related to the sodium channels, this is related to the leakage current. These are the probabilities for the gate to be open or activated in each ion, and these alpha, beta are computed are fitted from the experiment. So you see they are quite complicated values, but numerically it doesn't make any difference. It's very simple to integrate. I have 4 equations with some parameter, and then I have to fit also some numbers from the experiments. So with that I can model the neuron. What do I do with the synapses? Because neurons connect in a special way. How do I model the synapses? Well, the way we proposed with Raul at the beginning is considered as electrical synapses. But we know that in the brain, the majority of synapses are chemical and not electrical. So how do we model them? Well, first of all, this is the presynaptic part and this is the postsynaptic part of a neuron. When an action potential arises, it opens calcium ions here that enter, move some vesicles that contain neurotransmitters. These neurotransmitters are released and bind to the postsynaptic part and open new channels. So it's a complicated stuff. I have the wave coming, calcium enters, this vesicle moves, release some neurotransmitter, these neurotransmitters bind and open channels in the next neuron. So it's quite complicated, but in any case I can model that. But neurons are of 2 types. They are excitatory neurons and inhibitory neurons. If I have excitatory neurons, means that when I connect to another neuron, I will try to excite it. I will reduce the firing threshold. When I couple excitatory 2 neurons, I make the other one to be excitable. If I couple the inhibitory, I try to make it less excitable. So I'm increasing the firing threshold of the neuron. And those neurons are present in the brain, so I have to take into account both of them. So with this idea, how do we go to the model of Bose, to the neuronal circuits? So the idea is to propose that this circuit, that is more or less what Bose has proposed, is changed by this circuit. The master is a certain neuron that excitatory connect with a slave neuron, which excitatory connect with an interneuron, which is inhibitory, and goes back to the slave. So I have a circuit of 3 neurons to replace it, in which I'm not putting any explicit delay in the system. This inhibition will play the role of the negative coupling in the system. So this is the equation I showed you already. This is the Hodgkin and Huxley equation. This is the external current that I will put to all the neurons. And this is the connection. This is synapsis, which is this part of the neurons. This is accounted here. Certainly, I have to model the synapses. I told you the equation is quite simple. I have has a certain conductivity for the synapses. And then I have an evolution equation for the neurotransmitters and the receptor I have there, but I can model it quite easily. This is the kind of transmission, of connection, of function that connects 2 neurons, and then I have into account all that to be more realistic in terms of what happens in neuronal circuits. Now the question is, does it have any similarity what Bose has obtained? Well, this is the first case. I fix my excitatory connection between master and slave and interneuron. I fix that number. I fix my external input. And I change my inhibitory conductance between these 2. And then what I get for this value is I have the master making a pulse. I have the the slave making a pulse, and I have the interneuron making a pulse after, which is reasonable. This goes to this, this goes to this, this goes to this. This is what I expect. However, as soon as I increase this value, the situation turns around. Now, the second neuron pulses before the first does. So this is exactly what was found, that there is a transition at which the slave system is able to pulse before the master system. And this not only occurs for that, but you can do more numerics. And if you plot the delay time defined at the time at the slave pulses minus the time at the master pulses, I have a region in which, for different value of the injected current and different values of the excitatory connectivities as a function of the inhibition, I have a region in which I have the typical delay synchronization, the one I expect, but there is a big region in which I have anticipated synchronization. So this means that it is not so strange. This mechanism seems to be there. Actually, if I plot in the diagram excitation inhibition, I find three regions: one the delay synchronization, one the anticipated synchronization, and one in which the system does not synchronize, and we call a phase drift. And this is robust, again, external current, decay constant of synapses, driver neurons, and so on. So it seems to be a quite robust mechanism. But which is the mechanism that promotes? Why this happens? When Raúl studied that with me, he did a work with Marcena and other people in which they find that when you use the Voss scheme, that means that you have this coupling, which is a diffusive coupling, an excitable system, it has been proven that the excitability threshold in the slave neuron is reduced by the negative feedback, which means that if you have the two at the same level, you will never find anticipation. But if this is closer to the threshold, as soon as I receive a little information from the master, will fire. And will fire the other one. And you will see that clearer in some next transparencies. This is a figure in which they show what is the ratio between the slave and master threshold as a function of the coupling and for different delay times. And you see that if I increase the coupling and I increase the delay time, the difference in threshold is higher, which would explain why the second neuron is able to pulse before the first one. What happens in our case? Well, we have preliminary results that suggest that the system self-organizes. A situation in which, at the time the membrane current crosses the zero, which means that the neuron by itself is going to start to pulse, the inhibitory synapses, which come from other neurons, reaches a minimum value, facilitating the pulse in the slave. And that is what we see here. This is the membrane potential of the slave in dashed line. This is the membrane potential of the master. You see an anticipated regime already. This is the current in the membrane of the slave neuron. You see that it's coming almost constant, and then it crosses. At the point it crosses, the pulse starts. The pulse is developed because now I'm crossing the threshold. And look at what happened exactly at the same time, which I don't know, I don't see it, but there must be some lines here indicating that this line coincides with the minimum value of the inhibition. So at the time the inhibition is minimum, the system can make a pulse in the slave, and that pulse can be before or after the master. Okay, we understood something in a simple circuit, but with that you don't convince anybody, because if you don't do population dynamics, will not be enough. So I say, okay, let us extend these ideas to populations. So now I take a population, let's say 1,000 neurons, excitatory and inhibitory neurons in one population. I couple to another population, which is only excitatory neurons, and I couple this to a third population, which is only inhibitory neurons, following the idea of the three neurons I had before. So this is excitation, this is inhibition. We are not going to use the Hodgkin and Huxley model because it's too slow computationally. There are some other models that are good enough to be used, and one of them is the Iskiewicz model that describes the evolution of the membrane potential of each neuron and also contains the coupling with other neurons from the same population or from other populations. There is a second variable that describes the evolution of the ionic currents that are reduced to a single variable. And this, we suppose that we have synapses mediated by excitatory receptor and inhibitory receptors. These are related to what we call GABA receptor. These one are related to what we call AMPA receptors. Short interaction means in the same population can be excitatory or is mainly excitatory, but there are some inhibitory neurons here. Long range are known to be excitatory. And we include neuronal diversity, sparse connectivity, 10% connectivity inside the population, and also each neuron received an independent Poisson input, which means that if they are uncoupled, they will not synchronize at all. Okay, let us couple them. Let us couple among the population, in the population, and between two populations. And let us take some values for the master-slave connectivity, which in this case is 0.5 nanosiemens, and some connectivity between the interneuron and the slave population. And you see here that this is the master, this is the raster plot. Every time a neuron in the population makes a pulse, I put a dot. So every time one neuron in the master system makes a pulse, I put a dot. Every time a neuron in the slave system makes a pulse, I put another dot. And this is the interneuron. And you see here that the master one pulses before the slave one. But if I change the inhibition value, this one, in this case I reduce it, now the population in the slave pulses before the population in the master. So even in population, I can reach a situation for which the second population is anticipating the response of the first population. And you can see that clearer in what we call the local field potential or the main membrane potential. I just add all the potential of each population and take the mean value. And you see for the first case, you see here this is the master population in black, and in red I have the slave population. You see that it pulses before the master than the slave. But when I change the inhibition, then I go to a situation in which the slave one pulses before the master one. But if you look at here, the neuron is not at zero. There is some information that is being transferred from the master to the slave information. But that information is enough to make the slave population to pulse before the master. So there is not inconsistent. We are not violating any law of information transmission. If we go to the plane inhibition excitation, now we have two regions, one of delay synchronization, one of anticipated synchronization, and another region of delay synchronization. So it seems that we find regions in which anticipated synchronization is not so strange. But the question there at that time was, is there an experimental evidence? And I started with that at the beginning. I told you that there was some experiment from the group of Granger in which they observed already that when they recorded two areas in which one is sending information to the other, the second one pulsed before the first one. And they did that doing different measurements. They measured coherence and time delay and Granger causality. Coherence is the coherence function gives the linear correlation between two signals as function of the frequency. So in the Fourier transform, the autocorrelation of the Fourier transform of the time series of each population. The delay is estimated from the coherence spectrum. The delay is a certain phase which depends where the peak of the spectrum is, normalized to the frequency, and this phase is computed as the inverse of the tangent of the imaginary part of the spectrum divided by the real. So I can estimate the time. I can know how long it takes to go from one population to the other. Or at least I can know when the populations peak, when the action potential or the mean field potential is activated. And the Granger causality is a measure of the information flow. If a signal X is influencing another signal Y, then adding past values of the first variable to the regression of the second one, I will improve its prediction performance. So with this quantity, I can know how the information is flowing, from which area to which one. So I'm putting back one of the experiments by Bressler, and I put some dots, some arrows in color now. The red one indicates causal relationship with negative phase lag, in which the time lag and information flow do not coincide. In blue, we have the situation in which the Granger causality and the phase lag coincide, which means this goes to this and the time is positive. The activation time is positive between the two. And in orange, I put situations in which we have bidirectional connections. And in some cases, we have anticipation. In some cases, I don't have it. But I will concentrate, just as an example, in these 2 points, because they are quite clear. From here, I go to 1, and 1 activates before 2 does. So at that time we realized that we didn't need 3 populations. It is enough to play with inhibition inside the second population itself. So I have a master population which will correspond to the area 2, which contains excitatory and inhibitory neurons. I connect to the second population, which will be 1, which have excitatory and inhibitory neurons, and the interaction between the inhibitory neurons and the excitatory one have certain strength that I will change. I know from the experiment that site 2, Granger, causes site 1, which means the information is flowing from the site 2 to the site 1. And this is what you get from the experiment. This is the local field potential data for the slave in red, for the master in black. This is the coherence function, which has a peak at about 24 Hertz, in this case, which indicates at which frequency the population is oscillating, at which frequency it's oscillating, and allows me also to compute the delay time. And this is the Granger causality measurement, which indicates that the information goes from 2 to 1 and does not go from 1 to 2, which means this is clear, a unidirectional connection between 2 and 1. And this is the delay time they measure with this phase, minus 8.7 milliseconds, which indicates that this population, or the peak of this population, occurs almost 9 milliseconds before the peak of this population. And this is what we get from the numerical experiment by fitting the data. You see, this is our coherence function, which has a peak also at 24 Hertz, which corresponds to the experiment. We had a second peak, we couldn't avoid it. And this is the Granger causality measured from the numerics, which has to be, because in the numerics we are forcing the one direction. If we get here a wrong Granger causality, means that we are doing something wrong. If it is positive, as we find here, means that we are sending information from here to here, that what we obtain from the data is that we are putting in the model. And this is the delay time we are measuring, which is minus 8.2 milliseconds, which indicates that this situation represents what's going on in this experimental findings. Certainly, in the model we can do more. We can work at different frequencies that we know there are data, for instance, 7 Hertz. This is 18 or 7, or how many? I think it's 17 Hertz or something like that. This is 24. This point corresponds to the data from the PNS paper by Bressler. This point corresponds to the data of the Salazar et al. Paper, the science paper of 2012. Here we have the region for which inhibition promotes anticipated synchronization. Here is delay synchronization. If I go to this frequency, I still find a region of inhibition for which I get anticipated synchronization and a region for delay synchronization. And if I go to the PNS paper, I have 2 regions of anticipated synchronization I can play with. So if I take inhibition values around these numbers, I get anticipation. If I go here or here or here, I get delay synchronization. So it is broad enough. I can play with parameters and get situations similar to what they get in the experiment. Now the question is, what is the functional role of anticipated synchronization? And that's something we don't know. Actually, as far as I understand, nobody realized that this could be the mechanism that promote anticipated synchronization to occur, or the anticipated synchronization that is observed in the experiment. But we can speculate. For instance, we know that there are some situations, for instance this one, if you already are guessing which is the trajectory of the ball, so your brain has to act probably faster than the time it takes the ball to arrive you. So you are processing information at very fast speed. And there are some others also. It is known that in this case, if you are not fast enough, you will not be able to respond, because the time it takes the ball to go from here to here might be shorter than the time it takes to process the information, to react to that side. And this can be gained by learning, because we know that we are not able to respond, the serve, but these people are. And these people gain that because they have been trained. So the brain has been trained to accelerate the speed of reaction. And that might be related to the mechanism that we are finding. Still, this is a lot of speculation, but we can speculate more. There are some rules that are not in neuroscience. And one of the rules that is very accepted is the rule with respect to the plasticity of the brain. We know that the brain is very plastic and the connection changes with learning. And there is a rule which is called like the Herd rule, in which it says that the repetitive activation of one cell after another one leads to an increase in the synaptic strength between them. Which means I have a cell, is pulsing. The other one, which is connected, is pulsing. And if this one pulses first and this one second, the connection is increased. It is reinforced. However, if it occurs the other way around, if the repetitive activation of one cell is prior to another one, leads to a decrease in the synaptic strength. So if this one pulses first and this second, and this information goes in this way, I try to cut that connection because it's not useful. Well, that is what he proposed and has been found. So according to the Hebbian rules of the spike-time dependent plasticity, we have some result that can be useful. What means delay synchronization? Delay synchronization means that I'm reinforcing the connection between the two neurons, because the master pulse and then the slave pulse. So I'm in this situation. But what is the role of anticipated synchronization? Well, anticipated synchronization try to clean up synapses. And we know that during the day we accumulate a lot of things we see. We try to store a lot of information necessary to do that. But we somehow create some links that at some point has to be cleaned up because we cannot remember everything. So it might be possible that this mechanism of anticipated synchronization acts as an eraser of this memory or a cleanup of this memory for remembrance that you don't need to keep. Because at the end, what we remember, it seems to be storage in the connection between some neurons. But this is, I say, this is only a speculation.
But is this speculation in agreement with the previous one? I mean, if you say that this anticipated synchronization is useful for some things like catching the ball before
anticipated reaction.
Then if it is erasing thing, and if
no,
the ball is erasing, if the hand rule this erases this anticipating mechanism, on the next day you have to train again.
No, because
so it should be some balance somewhere.
Sure, there must be balance and they occur at different moments. Because something is what is trained with learning and has been established. And this can be a mechanism, but we don't know. We see this and has some relation, might have some relation with it. This is something that can be useful just to clean a little your hard disk every day. You don't want to keep all the information all the time. At some point you have to clean it. And this might be a mechanism that helps. We are not, this is only, we are physicists. This is only speculation. So in summary, we find a neuronal circuit of excitatory and inhibitory neurons that give rise to anticipated synchronization, the same we found more than 10 years ago with Raoul here, even in the absence of an explicit delay loop. The interplay between excitation and inhibition regulates the transition between delay and anticipated synchronization, that there are expressions of negative delay with positive Granger causality that has been observed experimentally in monkeys and are reproduced with the model. So this might be a mechanism for which this anticipation occurs. And if I am anticipating, what I'm doing is accelerating the information processing. So besides the reduction of information processing time or transmission time, any other functional role of anticipated synchronization is not clear yet. So that's all. If you want more information, you can find the other two papers, but most of the information is already included in Fernanda's thesis, and thanks for your attention.
So now it's time for questions. Yes, Luis?
So I'm going to better understand this anticipated synchronization. The textbook example of an AC circuit. Your current response can lead or lack the applied sinusoidal voltage. And there nobody talks about anticipation or because there you have a phase shift between the two responses, and that I mean the fact that the current leads or lag depends on whether the circuit behavior is capacity for inductance. And the circuit is linear.
Yeah, I know what you mean. And are you saying that that it's only a phase case? This is a matter of phase and not of time?
Yeah, I would like to know in your model why is reconciling to have this delay? Because in your model you have also this.
Well, in the model proposed by Voss, there was a delay. In the neuronal model, we don't include explicit delay. Delay occurs in the synapses itself. I cannot touch it. It's there. It's an internal delay that I cannot modify except if I change some neuronal parameters. But I'm not putting, in the neurons, I'm not putting an external loop with delay. I'm just putting an external loop of inhibitory neurons. That's all. But I'm not explicitly including any delay in the neuronal case. It's inhibition and the values of the parameter related to the synapses that gives rise to this delay. That I cannot avoid. I can change by changing some parameters. But what I thought you were asking, because many people say, well, but this is a periodic. What you are showing us periodic. So what is the delay, the time delay there? Well, it's true that it might be that it is not that one pulse is anticipating the other, but some pulse from the previous one is moving. Yeah, but it's just a phase shift. But then if you follow, as we did here, if you follow the transition, there is no discontinuity. So I don't think, if that is true, what you say would be true, and it might be possible, here, you don't see any discontinuity in the transition from delay to anticipated. If it's really a pulse that at some point jumps to the next one, it will be. And this has been proven also for chaotic systems, not only for periodic ones. So I mean, in chaotic system is clear the definition of time delay, which is not the case for periodic.
Yes.
But still, in the population, it's not perfectly periodic. So, I mean, here is quite periodic, but you can still distinguish the two pulses. No? So I think that here you can say, and we follow the transition, of course, and it is continued when crossing from delay to anticipated. There is no discontinuity there. So our guess is that it's really an anticipation in time. And that is what they have also measured in the experiment. They have also found in the experiment.
Have you tried to measure causality with transfer entropy, not just with Granger causality?
Not yet. We are measuring causality with complexity measurement, which you remember that Osvaldo Rosso proposal of measuring complexity with embedding things. You can involve a series with time delay, and they are doing right now some measurement, and they are finding the negative delay there. And I also proposed Victor Lopez to do some transfer entropy measurement to see whether this also reveals the same sign for the time.
And another question, could it be, for instance, when we put the example with the connection between region 2 and 1, could it be that there could be another region hidden?
Sure.
That makes
sure.
There's a negative time delay?
Sure, it can be. In the brain, in real experiment, you are never sure about anything, because the circuits are so complicated. That's why we took exactly that one, which is the clearest for us, which you see a clear one directional, but if you see before, there is also some other influences. So we cannot be sure about that. But I mean, at least where was it? Or here. There was, you see here, there are some other interactions with other areas that we are not accounting. But maybe it can be done some more sophisticated analysis could be done with partial coherence or partial transfer entropy. That could be done, but we didn't do that yet. Actually, we used the same technique that they used in the experiment. Our aim was to show that there exists a mechanism that could explain those observations, not to do something new. And the referee said the same. Why don't you measure this? Because we are not trying to do new indicator measurement on the data. We try to see that what they observe in the data can be explained with the model. That was all.
I mean, my question goes a bit along the line of the first comment. So you use Poissonian statistics, so you have a periodic drive more or less.
Poisson noise. It becomes periodic due to the interaction.
The Poissonian term in your, where was it?
It's in the voltage. No?
So it's in the voltage, so it's a rather periodic function.
Why?
Naturally.
What do you mean
Yeah,
periodic?
no, go back.
It's here. It's about here. This term contains the synapses and the external forcing. And the external forcing is each neuron receives an independent Poisson input due to the interaction they synchronize.
And Poissonian is then more or less periodic in time. So
No,
for a single photon
no, you have an average
no,
time,
no,
but
no,
it's not.
not a single photon source. You have Sony statistics. So on average, without short noise, you get perfect temporal sequence.
But that is not what you generate numerically. Numerically, you generate a distribution which is not periodic, which gives rise to train of pulses that is not
But you
periodic.
see in your response system that is rather periodic.
That is because of the interaction. They receive independent Poisson law, that due to the interaction they are able to synchronize. But if you see in the raster plots,
No, but already
look at the distribution of pulses. They are not pulsing exactly at the same time.
No, but the overall system still follows the rather periodic temporal.
Well, there is a rhythm. We know that the population have a certain rhythm always, but this rhythm is created because of many neurons pulses at slightly different
No,
times.
but this is not the rhythm which is also present in your Poissonian external driving. So my question is rather because if you then approximate the system as a harmonic, as a linear oscillator, if you change the parameter, you would also not necessarily get a discontinuity in your phase. You would get a continuous shift, no? And then I could also imagine a system like this anticipated and delayed.
We can discuss
Well,
it. I'm not sure.
Anticipated.
I have another question. Whenever you show the dependence on the coupling strength in the inhibitory.
This one?
No, right next.
Tell me.
Which one? No.
This one?
No, okay, this one. Is any way to understand why is this non-monotonous form? Why there is two ions of negative synchronization? Why this have this dependence? Is any qualitative explanation or?
Not really, because it's so complicated. You have 1,000 neurons, all of them different to each other, all of them receiving different input, and they are interacting within them. So look at this, and here I only changed the Poisson rate to generate a slightly different rhythm here. I have here at 17 hertz, I have here at 24 hertz. So the only difference I'm inducing a
You
different
don't
rhythm.
change the coupling strength, nothing. I mean, between the two places, you don't change anything.
No,
Just change the
just change the rhythm of the internal population. And then you can get different results. That is, yeah. No, and certainly we are trying to also understand a bit more in the population if we get the same mechanisms as we got for the 3 neurons case, which you see this self-arrangement of the current, the membrane current and the synaptic current to cooperate to generate this pulse, and we were not able to find it yet. With the population, it's very complicated because you have a lot of things that are difficult to see.
And within a population, how they are numbers coupled? You randomly?
Yeah, you take 1,000 neurons, you connect 10% of, you take one neuron, connect of 10% of the neurons, 100, and 80% are excitatory, 20% are inhibitory, and that's all.
Randomly.
Randomly.
Without any structure.
And that is the receipt that people give in computational neuroscience to build, let's say, cortical areas, cortical circuits.
I'm not sure whether I grasp the meaning of Granger causality, but anyway, you mentioned that it can go in one way, the other, or in both.
Yeah, you can have the same value of Granger causality in both directions. You measure a Granger causality value. You say this influenced this one, and you get a value. And then you say I will see if this one influenced this one, and you get the same value. Then the relation is bidirectional.
Okay, but it's not temporal sequence. It's not the first one causality occurs in one direction,
No.
then another.
No, not necessarily. Actually, you are taking, if you take a long time series, you don't know if they are shifting in time, for instance. I don't think you can distinguish that. I'm not sure.
Any more questions? No? I think that we can thank you.